Hello, everyone. I'm Jeannie Linky Northrup, and I'd like to welcome you to today's live broadcast, Lipid Formulation Made Easy with ABISOL, sponsored today by ABITEC and IMCD Pharmaceuticals. In today's webinar, you'll learn more about the advantages associated with lipid formulations and how the ABISOL kit addresses lipid formulation complexity. Lab case studies that use the ABISOL kit to generate innovative formulation concepts will also be discussed. ABITEC is a world leader in the formulation, manufacture, and supply of specialty lipids. With an expansive portfolio and experienced technical experts, ABITEC is able to develop solutions for the pharmaceutical, nutraceutical, and specialty chemicals market. ABITEC delivers solutions in solubilization, emulsification, and lubrication across the globe. IMCD is a leading global distribution partner and formulator of specialty chemicals and ingredients. With a global network of technical centers, IMCD develops bespoke solutions to support every stage of product development. They serve as an end-to-end partner in the pharmaceutical, nutraceutical, biopharma, synthesis, as well as the agrochemical sectors to advance ideas for a healthy future. There are just a few important announcements I'd like to make before we begin with today's webcast. First and foremost, we have two great presenters on who are eager to respond to any questions you may have about their presentation. We encourage you to submit questions to them by typing them in the Q&A box that can be found at the bottom of your video player. You can also enlarge your slide window by clicking on the small icon in the bottom right corner of your media player. Please note, however, that all slides will advance automatically throughout the event. If you do experience any technical problems viewing or hearing this presentation, please click on the question mark help widget located in the top right of your presentation window. I would now like to introduce and welcome today's experts and our guest presenters, Drs. Heath Bowers and Chhanda Kapadia. Dr. Bowers joined ABITEC in 2018. Dr. Kapadia actually joined IMCD in 2018. Dr. Bowers currently serves as the North American Technical Business Director for Pharmaceuticals. In his role, Dr. Bowers assists formulation scientists in the development of lipid-based drug delivery systems. Additionally, he has contributed to academic as well as industry collaborations, new product development efforts, and application research to further ABITEC's place in the global lipid excipient market. Dr. Kapadia serves as the Technical Director of Pharmaceuticals for the Asian Pacific region. She is adept at technical service and troubleshooting and application laboratory setup. She formulates marketing strategies and technical promotion plans for new business generation and leads efforts from original concept through implementation to handle a diverse market dynamic. She has been leveraging technical expertise in the field of pharmaceutical excipients and has a strong scientific background. So Drs. Bauer and Dr. Kapadia, welcome. Thank you so much for joining us, both of you today. Dr. Bowers, I believe you'll be kicking off today's presentation. So please feel free to begin when you're ready.
Thank you, Jean. So I want to thank everyone for joining today. To begin, I want to explain kind of the fruition of how this all came about. ABITEC, during my 5 years with ABITEC now, I visited multiple formulation scientists and have come across many different conversations and challenges that they all arise. Most of these challenges generally, as expected, are around solubility and absorption of APIs. And it's not unexpected. 88% of novel therapeutics that were approved by the FDA in 2022 suffered from low solubility. A majority of these were BCS2 molecules. There was also BCS4 molecules, but there were a bunch that were not defined in the BCS class, but they suffered from low solubility and high log Ps. Now, there are a lot of different ways to handle low solubility, and I'm not going to say that there's one way that's better than the others, but being a lipid manufacturer, we tend to like lipids. Lipids can completely dissolve hydrophobic APIs because they themselves are hydrophobic. And what they do is they encase this API in a fat bubble. And as a result, they prevent the API from being degraded from hydroxyls or peroxides or anything of that nature. They can also provide an extended release profile as a result of being encased inside a fat bubble. The other nice thing about them is, they will completely emulsify and highly disperse your API in the GI tract because of the nature of the chemistry. The other nice thing is APIs are natural. Your body knows what or not APIs, I'm sorry, lipids are natural, and they know what to do with them, right? And that's why they're generally recognized as safe by the FDA. Your body absorbs lipids on a regular basis, and if your API is, encased in lipids, it's going to go along for the ride as well. So there are multiple types of lipid drug delivery systems. The one we're going to talk about and focus about today is self-emulsifying drug delivery systems. These are very simple systems in the sense that you just combine a 3 or 4 different types of products. So you have an oil, an emulsifier, a surfactant, and possibly even co-surfactant to create a preconcentrate. This preconcentrate then allows for the drug to be dissolved in it and then become solubilized, right? Once this system hits an aqueous environment, you see they will completely emulsify and create vesicles. These vesicles will then encase the API and prevent them from coalescing and precipitating out a solution. Some of the benefits of the self-emulsifying drug delivery system is that, as I explained already, they increase solubility of your API. So as increasing your solubility, you're going to increase the ability of your body to absorb that product, leading to higher bioavailability. So, and you may have a couple situations where your API is also not very permeable to the GI tract apical layer. So you may have some enhanced permeability as a result of being encased in the lipids as well. We've already talked about how these fat bubbles can hide your API, so to speak, from things like peroxides and hydroxyl groups. They're also very amenable to various dosage forms. Anything that can handle a liquid can handle a SEDS formulation. So, as you can have a soft gel or a hard gel, you can dose it in the form of an eye drop or some type of injectable. So there's really no limit to where SEDS systems can go in terms of the dosage forms. And the last key feature here is that SEDS forms are actually very easy to manufacture on a large scale. You're basically mixing, 3 to 4 liquids in a vat and blending them to homogeneity. So it's not a very difficult commercial manufacturing procedure to go to create one of these on a commercial scale. And I've already mentioned a little bit about what a SEDS formulation looks like, but picture it as more like a micelle, right? You have a hydrophobic core, which then is surrounded by emulsifiers and surfactants that bridge the gap between the hydrophilic nature of the GI tract and the hydrophobic nature of the hydrophobic core, which is typically an oil. These micellular systems are very thermodynamically stable. If they're done right, a SEDS system can be emulsified indefinitely. And as I mentioned, these generally consist of minimally some type of lipid or multiple lipids, and then a surfactant, and then of course the aqueous environment which they're going into, which would either be the stomach acid or the intestinal acid or intestinal fluid. And SEDS systems have been around for a while, and so as you can imagine, they are widely used in commercial products. There's a whole host of therapeutics that I've listed here that all use self-emulsifying drug delivery systems. Anything from testosterone to oral peptides like octreotide. You can even see that there are some that treat hepatitis C viruses. So there's a whole lot of indications as well as types of APIs that can benefit from a self-emulsifying drug delivery system. And these are just a few that I've mentioned here. However, with every great thing, there must be some challenges associated as well. And such are not necessarily the most quick formulation approach, if you will. They typically are a multi-step process, which requires doing an extensive solubility screen. So you generally would screen multiple types of oils and surfactants and emulsifiers to try and figure out which one provides the best solubility for your API. Once you've done that, then it's a case of doing a pseudo-ternary phase diagram. And really what that is basically a methodical approach of trying to find the most stable emulsion possible, where you will start with a pre-concentrate mix of oil, surfactant, and emulsifier, and then start diluting it with water to see how much dilution you can get, how diluted you can make this pre-concentrate, and it still creates a self-emulsifying drug delivery system. Of course, once you've done all of this and you've found a pre-concentrate that works the best, you can then do a dissolution study and followed by pharmacokinetic and pharmacodynamic studies. That is what you would do with any type of formulation. But it's really the first two that kind of take a long time and a lot of effort because it's very tedious and laborious. So after, visiting several formulation scientists over the years, what I found is that, everyone agrees that SED systems are by far the best way to deliver an API. However, it's just not really a feasible approach when you have, say, you're a discovery scientist and you have your organic chemist giving you 100 molecules in a week, or maybe you're given only 10 milligrams of material and screening multiple lipids and multiple surfactants and emulsifiers is really not feasible because you don't have enough material. So as a result, the majority of the formulation scientists that I work with, they'll take just one oil. Maybe they'll screen like 5 different oils and figure out which one does the best. And then they'll dose it directly as that. They'll directly dose it as a neat oil formulation into a rat and see if they can get some type of pharmacokinetic absorption. But generally, this is not a good approach because what will end up happening, especially if you're using just pure surfactant, is the API, once it hits that GI tract, will then precipitate. It'll end up precipitating out and you won't get any absorption and therefore you won't get any bioavailability. So it ends up being a waste of time and effort on the discovery scientist's approach. So we really thought, okay, there really needs to be a solution that handles this, gives you the benefit of the self-emulsifying drug delivery system, but offers it in a very easy to use way. And that's where we thought, okay, well, we need a set of pre-concentrates, SEDS pre-concentrates that addresses all this. But we didn't want these SEDS pre-concentrates to have some type of random rhyme or reason kind of approach. So we thought, let's vary the hydrophobic lipophilic balance, or the HLB, of each and every single one of these pre-concentrates such that you have one pre-concentrate that is highly hydrophobic and one pre-concentrate that is highly hydrophilic. And then you have a range in between this two. That gives you an approach to try and screen only, let's say, 5 or 6 different types of pre-concentrates instead of screening multiple different oils and trying to create your own SEDS pre-concentrate. Now, we don't want these pre-concentrates just to be any type of emulsion. We want them to be type 3B emulsions. And the reasons why we want type 3B emulsions is they have very small particle sizes, somewhere between 50 to 150 nanometers. So that's ideal. And to get that, you typically have to have a high surfactant concentration. So anywhere from 40 to 70% surfactant. What that does is it allows for an easily dispersed emulsion that sets up without any high energy. So it's very low energy. You literally take the pre-concentrate, put it into an aqueous environment, shake it, and it's emulsified. So to do this, we decided, all right, we're going to use, of course, our own excipients, and we used Captex 300, which is a medium chain triglyceride. We used our Capmol MCM, which is a monodiglyceride, and then we used 2 surfactants. And the reason we used 2 surfactants is because we wanted one that had a very low hydrophobic lipophilic balance, and then we wanted one that had a very high hydrophilic lipophilic balance. So that way you could vary the HLB for all of these preconcentrates. And so the Achenon CMG6 is our low HLB, whereas our polyoxyl 35 castor oil is the high HLB surfactant. We created a design of experiment, or DOE, to go about creating these, and the response elements for the DOE was based off of HLB as expected, but we also wanted particle size to be a key feature of this. So those were our 2 response elements for the DOE. When we performed this DOE and did all the results and then put it back into the program, we got great R2, we got a great adjusted R2 value, but our predicted R2 value was a little bit off, which was 76.8. And we were really perplexed by that. And what we found out was that it was fact that the predicted was a bit off was due to the fact that Capmol and Captex have to be held below a certain ratio. If you have too much Capmol, which is our monodiglycerides, in relation to our medium chain triglycerides, so too much of the mono versus the tri, what ends up happening is the vesicle size balloons. So instead of it having a very small particle, you end up having a much bigger particle because this ratio is off. Once we determine that, and we actually set within our parameters that these 2 have to be within a certain ratio, we were actually able to get a predicted value of 99.9 again. So we were able to adjust all of that. Once we had that, we went ahead and took the data and created 5 preconcentrates, with the lowest HLB being around 5 and the highest HLB being 10. So we have a very low hydrophobic preconcentrate, and then we have a very high hydrophilic preconcentrate, which is around 10. And then we had a range in between those. When you look at the characteristics of these emulsions, the preconcentrates are very, very good. So the particle sizes are all about 55 nanometers or smaller at a 1% weight emulsion. And what's better, not only are these particles very small, their polydispersity index is incredibly small. Typically, for a PDI, you would want a value somewhere around 0.25 or lower. Our PDIs, or polydispersity indexes, are about 0.1 or lower. So what that really means is the population of particles are all pretty much the same. They're all about that 50 or 5 nanometers or smaller. And the other great factor there is that the polydispersity index refers to how stable the emulsion is. So the lower, the better. So having a lower PDI really states to how stable the emulsion is as well. We did do a couple, proof of concept studies, and you'll see here preconcentrate number 5 solubilized ibuprofen the best. And now we know that ibuprofen is a BCS2. It's has a log P of 4, so we thought it'd be pretty hydrophobic, but in reality, the most hydrophilic preconcentrate ended up solubilizing the best. And we really believe this to be the case because of the carboxyl group at the end of ibuprofen. We know ibuprofen is actually soluble in the intestinal tract, so pH 7, so to speak. It's just really not soluble at under acidic conditions. So really, I think that carboxyl group is interacting with the hydrophilic nature of the preconcentrate number 5, which is why it's more soluble in ibuprofen, or in preconcentrate number 5. The other key feature here is that once we take ibuprofen and put it in preconcentrate 5, the particle size still remains small. I don't know if you recall, but the preconcentrate number 5 by itself with no ibuprofen had a particle size around 22 nanometers, somewhere around there. Now that we've added in ibuprofen, the particle size is only increased by 56 nanometers. So it's gotten a lot. It's still incredibly small. And to give you an idea how small this is, delivering drugs to the back of the eye is very, very difficult. To do that, you typically have to have a particle size less than 90 nanometers. This is a 56 nanometer particle size. You could theoretically deliver ibuprofen to the eye through an eye drop and get immediate headache relief. So, this is something unique to these preconcentrates. And while I'm not suggesting someone create an ibuprofen eye drop, it is something that could potentially be done. We did do a couple other products. I just want to show you this last one here, Sonarazine. We really didn't have a great analytical method to determine the concentration in each one, but what I do want to show you is the particle sizes that we obtained with Sonarazine. Every single one of these, and this was done in triplicate, every single particle size within the ABISOL 1 to the ABISOL 5 are incredibly small. You're talking, again, 50 nanometers or smaller with the Sonarazine inside the particle. So this is a very, very consistent result that we're getting, where the particles are remain very small. And you can see that a little bit closer here, where you see is ABISOL 1 has a particle size around 40, and ABISOL 5 is all the way down here at 20. Again, the PDIs are 0.1 or lower, and you can see how tight some of these PDIs are. Very, very, very small. So before I wrap it up here and hand it over to Chhanda, I want to explain some of the things that we've seen in the market since we launched ABISOL. We've had a couple customers perform intramuscular injections with ABISOL 2. The study was done in rats, and it was very, very successful. They saw no adverse events. They have studies planned for canines as well. We have a CDMO that is now using ABISOL as their first screening attempt. So when they get a new API in and they need to get it solubilized, they pull ABISOL right off the shelf, and that's their first go. We also had a client who had a log P12 molecule. So the molecule was log P12 and had a greater than 1,000 moles per gram molecular weight. So a very large API with a very high log P. They tried multiple different things. They tried cyclodextrin, they tried various types of micronization. They could really not get this thing to solubilize. And what they found is that ABISOL was the key, and they were actually able to move forward with studies as a result using ABISOL. So really key here is that ABISOL may not be the end all be all for you moving forward, but it may be a great start for you to have in your toolkit when you get new APIs, because it'll certainly solubilize the majority of your APIs. And with that, I'm going to pause and turn it over to Chhanda, where she's going to actually discuss her results using the ABISOL kit with various APIs. Chhanda?
Yes, thank you so much, Heath, for this very informative and lucid presentation. And it aptly correlates to the title of Lipid Formulations Made Easy with ABISOL. So thank you so much for that. And I wish a good morning, good afternoon, and good evening to all our participants in this webinar, and a warm welcome to all of you all. So post presentation from Heath, what I'll be taking you all through some of the case studies and some innovative formulation development that we worked out in our lab in Mumbai, India. And this was worked by the IMCD technical team of India. So let me quickly take you through the agenda of my presentation. So we've understood on ABISOL. What we've done in the lab are certain case studies where we have evaluated solubility of APIs in the different ABISOLs. we've also tried to establish a correlation between certain physicochemical properties of the APIs to the ABISOL different ranges that we have in terms of HLB. Also, what we've done is try to evaluate the scope of ABISOL in formulation development. As Heath rightly presented, that ABISOL is amenable to different formulation developments, whether you talk about tablets, capsules, ophthalmic solutions, injectables, or even topical formulations. So we'll touch upon that as well, where we find the scope for evaluating ABISOL. And one small case study, which is still in the process at our Mumbai PTC, where we've done development of a very challenging loratadine ODT with ABISOL. So I'll take you all through that as well. So moving ahead, this is just a quick summary of what Heath presented, so I'll not be going into that, but you'll have to understand that the composition of ABISOL is really very, very simple. You have Captex 300, you have Capmule MCM, you have Aconone CMG6, and you have polyethoxylated castor oil. And you have ABISOL of 5 types with different HLB values as explained by Heath. So what we've done is that in our lab, we did the solubility evaluation of different APIs. Now, these APIs were selected basis their log-P values, basis their molecular weight, their structure, and also their melting point. So all these parameters were taken into consideration, and a group of 17 APIs were selected, and their solubilities were evaluated in the different ABISOLs. So what you see highlighted in purple gives the solubility, the high solubility levels in the ABISOL range. And what you see in highlighted in blue is which particular ABISOL works as a potential solubilizer for that particular API. So if you see these APIs, you see that there is a considerable high solubility for certain APIs. You see it for fenticonazole, you see for clotrimazole, you see a very high solubility achieved with fenofibrate, with loratadine, with carbamazepine. Some surprises we also observed were for a high solubility for paracetamol as also for salicylic acid. So taking this data into consideration, we tried to establish a correlation. And we would like to bring these critical observations, what we observed, to your attention, which could help in formulation endeavors or solubility endeavors that you all take up in your R&Ds. So to bring to your attention these correlations, what we observed with the API properties are, so when we worked on the different APIs, like especially when you look into loratadine and fenofibrate, so these are, we found that they exhibited excellent solubility in ABISOL 2 and 3 because of their high log-P values. So these are more lipophilic in nature. And as the HLB of ABISOL increased, their solubility decreased. So that could be the reason, because the log-P value is high, they are more lipophilic in nature. They were solubilized well in the lower HLB values of ABISOL. The surprising observation that we found was with paracetamol and salicylic acid. So if you see their log-P values, the log-P values are low. And they are more, of course, they are hydrophobic, but more of the chalk powder kind, not the greasy lipophilic molecules. Now, here again, we found that they exhibited good solubility in ABISOL, and that we thought that could be attributed to the functional groups present in these particular APIs, especially the hydroxy functional group, which could hydrogen bond with the ABISOL, and that could enhance the solubility. Moderate to low solubility in ABISOL range was found with certain APIs like carbamazepine, budesonide. So if you go to see per se, the budesonide solubility, if you go to see per se in ABISOL, is low. But we have to take into consideration that this is a low dose, highly potent API. And that worked as an advantage because that was good enough to solubilize budesonide and deliver it as a solution, because as such, the dose of budesonide is less. So moving ahead on this, when we try to just summarize all these parameters or these observations that we observed, is that ABISOL works as very good potential solubilizers for APIs that could have OH and NH groups, which could facilitate in forming hydrogen bonds and help in better solubility. So the target APIs here were paracetamol and salicylic acid. It also works well for the free form of the APIs rather than the charged APIs. And this particularly was observed for minoxidil because minoxidil exists in the charged form and the solubility of minoxidil in ABISOL was less. As I mentioned a little while ago, it can be evaluated, ABISOLs can be evaluated as a solvent for very potent APIs like low dose APIs like budesonide. Also, low to moderate melting APIs facilitate solubility in the ABISOL range. This was also one of our observation, taking fenofibrate into consideration. Depending upon the molecular structure, so if there is a lot of steric hindrance in the molecular structure, like what we saw in budesonide, it results in a decreased solubility in ABISOL. Worthwhile to evaluate ABISOLs as solubilizers for high molecular weight lipophilic API. So here again, fenofibrate falls into that category, loratadine falls into that category. Now, this is to understand that we evaluated almost 17 APIs. And we feel that this is an inadequate sample size, and we really need more number of APIs to establish an exact correlation between, say, the log P value, the molecular weight, and solubility. Having said that, these are some of the critical observations which could help you all in your formulation or research endeavors in your R&Ds. And if you all have any APIs where you all feel that there's a potential to explore ABISOL, please feel free to reach out to us. Now, moving ahead and understanding the scope of ABISOL in formulation development. So it generally comes to a mind that these are lipids, these are lipophilic liquids. So how would it be possible to convert them into an oral solid dosage form? So that's always triggers a formulation scientist. But really, it's very, very simple. We worked out some case studies in the lab, and we can confirm that it's not very difficult to get into the liquid solid technology with lipids. So definitely, we are all aware that SMEDs and microemulsions, they improve the solubility, bioavailability, and can be evaluated as to be filled in soft gel capsules to be evaluated as oral solid dosage forms. Also, as tablet capsules, solvent based in liquid solid technologies, we can get into that working. And of course, as oral liquids, as emulsions, suspensions, drops, concentrates, ABISOL can be evaluated. Another important area where we can really focus with ABISOL on is in topical dosage forms. So it can be a part of creams, lotions, ointments, because it's a combination of different surfactants, oil. So it could act as solubilizers, could also act as an emollient in a topical formulation. Moreover, if we are thinking of working in a clear gel formulation, it can work as a solubilizer in a clear gel formulation, because on dilution with water, as Heath presented, the clarity is retained. It's a clear formulation, so definitely it can be used in topical gels. And moreover, in topical dosage forms, when one thinks of using microemulsions, the cost parameter always comes into picture. But definitely with a topical dosage form, we can go in for a localized kind of a drug delivery by using spray formulation. So it could be a topical spray, it could be a topical microemulsion foam. So in those kind of systems also, we can evaluate ABISOL. So you can see the broad scope in formulation development which is possible with ABISOL. Now, ABISOL as a pre-concentrate to troubleshoot numerous challenges. So these are some ideas which we are suggesting to you all, and which can really help you all in your formulation development. So one thing which comes to our mind is how could we go for dose reduction of APIs, which is very, very important, because many of the APIs which are available in the market are poorly soluble. They have a variable bioavailability, owing to which you generally end up administering a large dose, resulting in severe side effects. So improved solubility of BCS class 2 and 4 drugs with ABISOL will, of course, we can go on to reduce the dose and ultimately reduce the side effects. And a classic example which we did in our lab is solubilizing fenofibrate, where we observed a very good solubility of almost 92.47 milligram per gram in ABISOL 2. And the dose of fenofibrate is in the range of 90 to 200 milligram per dose per unit. So there is a possibility of improvement in solubility and dose reduction for fenofibrate with ABISOL 2. Now, this has worked for fenofibrate, but it can be a platform technology for APIs like fenofibrate, wherein we can go in for dose reduction. So this is one idea which we would like to bring to your attention. The second one which came to our mind is impurities reduction. So when you talk about oral liquids, or you talk about pediatric oral drops where there are concentrated solutions of APIs, there is a need for solubilizer in oral liquids. The very, very commonly used ones are propylene glycol, glycerol, which are added as co-solvents or solvents to these kind of formulations. And now we've seen a lot of case studies coming up across the globe wherein impurities associated with these co-solvents have led to a lot of formulations being withdrawn from the market because of the presence of these toxic impurities. And here stands the strong advantage of lipid offerings like ABISOL. So what we saw that there was a good solubility for paracetamol in ABISOL 3 and 5, almost 50 milligrams per gram. So when one is talking about a pediatric formulation, where up to almost 15% of propylene glycol is used, why can't we think of replacing that with ABISOL? So ABISOL works as a very good alternative to these kind of solvents which have a high risk of toxic impurities. So this is one suggestion which also comes to our mind. And of course, for solubility enhancement, when one talks about topical formulation development, so here we observed a good solubility for one of the APIs, that is apremilast. So apremilast is an API used for the treatment of plaque psoriasis, and what's available in the market is an oral formulation of apremilast. So apremilast stands up as a very high potential API for topical formulation development. And with this good solubility in ABISOL 4, definitely ABISOL 4 can be a part of developing topical formulations. Could be a topical cream, could be a topical gel, could be a topical lotion, could be a topical foam formulation, wherein ABISOL can function as an emollient because it's a good solubilizer. It could also help in enhancing the permeability of apremilast across the skin. Because of the emulsifying base concentration of ABISOL, it can help in giving stabilization to the end formulation, and all this devoid of any toxic impurities. So there stands a strong rationale for promoting ABISOL in topical formulations as well. Also, as mentioned earlier, solubilizers for low-dose APIs like budesonide, which exhibited a solubility of almost 8.85 milligram per gram in ABISOL 2. So, possibility of exploring ABISOL in extremely low-dose, highly potent APIs. Continuing further, ABISOLs can also function as crystallization inhibitors for high-dose APIs. So solubilities of actives when present in high dose are prone to recrystallization. So generally it results in precipitation of saturated or supersaturated solutions on storage. So your ABISOL finds a very good scope for evaluation, because we did observe it for salicylic acid, where salicylic acid, because of the presence of the functional groups, exhibited good solubility in ABISOL 2. So ABISOL can be incorporated to replace certain organic solvents which are used like isopropyl alcohol to solubilize salicylic acid in topical formulations. So we also see a good scope for ABISOL in acting as a crystallization inhibitor for high dose APIs. So quickly taking you all through the ABISOL case study that was executed in our IMCD Mumbai PTC, where the objective was to develop an orally disintegrating tablet of loratadine with increased solubility and evaluate ABISOL as a system that provides micellization and helps in better dissolution and solubility. So this study is in the process. We have still to complete the stability aspects of this study, but we thought why not present this study to give you all an idea about how one could work with ABISOL and formulate an oral solid dosage form. So this is a formula card that we have worked with loratadine, solubilizing it in ABISOL. This was worked at a 1:1 ratio. And we evaluated calcium silicates as adsorbents. Now, calcium silicates worked really very well, and almost one part was able to absorb 6.6 parts of the loratadine ABISOL mixture. This was further blended with other ingredients to make it into ODT and suitably flavored as well. So if you see the manufacturing process, it's really a very simple process where loratadine was solubilized in ABISOL, and this mass was then adsorbed onto calcium silicate, which resulted in a free-flowing powder. This was blended with spray-dried lactose, suitable disintegrant, flavors, lubricants, and compressed into ODTs, which really resulted in quick disintegration. We were able to achieve a good hardness, friability as well for these ODTs. And of course, we are in the process of evaluating the stability of these formulations as well. So just to quickly give you a schematic representation of this, most of the pipeline drugs are with low solubility. So introducing ABISOL as a complete lipid kit, which will help in solubilizing APIs like loratadine and improving the tabletability of this with Zeopharm 600, the calcium silicate, which can be evaluated as an adsorbent, and blending it with suitable ingredients like spray-dried lactose, resulting in quick disintegration time. So here we could very well evaluate the scope of ABISOL in a formulation like ODT, which is quite a challenging formulation because you need to achieve the disintegration time of less than 30 seconds. And we were able to achieve with ABISOL and loratadine. So summarizing and bringing to your attention is the fact that many of the marketed drugs belong, almost 40% of them belong to BCS class 2 and class 4. And the CAGR for solubility enhancement excipients has always been increasing. So you can understand the demand which is there in the market for solubility enhancing excipients. And almost 80% of the NCEs and pipeline drugs are with low solubilities. And thus, ABISOL works as a versatile tool for solubility enhancement. So it's not only enhancing the solubility, but also the scope for ABISOL in different formulation developments, like it could be tablets, it could be soft gels, it could be hard gelatin capsules, it could be topical dosage forms, it could be ophthalmic solutions, injectables, and so on. So it's really lipid formulations made easy with ABISOL. So thank you for your very patient listening, and thank you to everyone. And I would especially like to thank my technical team at IMCD India. So Dr. Vivek Dhawan, who is the technical manager for IMCD India, Prajal Pandhare, who has worked on this project in the lab, and he's a lab manager, and Rahul Yadav, who is also the lab manager, and he's supported on the solubility studies as well. And thank you, Heath, for collaborating on this webinar. So over to you, Jenny. Thank you.
Thank you. Thank you so much to both of you. That was a fantastic presentation. A lot of significance for looking at lipid-based formulations. I've seen a lot of great questions also populating in throughout the broadcast from our audience. But before we do jump into the question and answer session, I just wanted to remind our audience, for anyone who may have signed in a bit late, how you can submit your questions to Drs. Kapadia and Bowers. You can fill them out in the Q&A box on your screen. That could be found directly below your video player. I'd also like to let our audience know that I'm seeing quite a few questions come in, so I want to be mindful of time and let you know that any questions that the doctors do not have a chance to respond to live today, they will be reviewing and responding to offline as well. So please do not be discouraged to submit questions, even if we start running a bit low on time. So I'm going to pull up our questions for our panel. The first question I have, how reliable are the HLB data ranges?
I think I can take that question. So with regards to the hydrophobic lipophilic balances, those are all based on the emulsifiers that we use within the, and surfactants that we use within the within this preconcentrate. And we did this mindfully in the sense that we created the design of experiment that calculates the HLB based on the percentages of those emulsions or those surfactants and co-surfactants that are in there. So as long as we make the preconcentrates the same every single time, the HLBs will be the same every single time. So they are pretty reliable to answer the questions in short.
Okay, great. And can ABISOL be used for improving solubility with hydrophobic peptides?
Yeah, and this might be a question that both Chhanda and myself answer. So the ABISOL kit can definitely be used with peptides. Good examples of this already in the market, not necessarily with ABISOL, but with SED systems, is neo-oral or cyclosporin, as well as voclosporin. Both of these anti-inflammatory peptides are used, or rather use SED systems to deliver the hydrophobic peptides. So you could certainly use these preconcentrates to figure out which one works best for your hydrophobic peptide and deliver it in the same way. And I don't know if Chhanda has anything to add there.
Yes, Heath, I do sync with you on that. Definitely, ABISOL can be evaluated for hydrophobic peptides as well. Of course, they need to perform the solubility studies and find out which ABISOL works the best, and it certainly can be evaluated. Yes.
Thank you. And Dr. Bowers, you were mentioning SEDS. So the next question I want to jump to is talking a little bit about ingredients. What is the shelf life for any of the SEDS ingredients?
Yeah, so the individual components all have shelf lives that vary. We have tested the shelf lives of the pre-concentrates themselves. So far, we've tested up to over a year. Actually, we have an 18th month shelf life coming up, study coming up here very soon. So I believe it'll probably be stable at least to 18 months because one of the ingredients in the pre-concentrates has an 18-month shelf life. So it's probably going to be around 18 months or more, but it'll depend once we do these studies.
That's great. Thank you. It's pretty significant in terms of time. What about animal toxicities? How high have you been able to go in IND-enabling animal tox studies?
And again, this is a great question. It really, I mean, what we've seen in the literature is you have to keep in mind these preconcentrates are made with compendial excipients that are already being used in therapeutics on the market. So while we haven't done, let's say, studies using preconcentrates directly in animals ourselves, we have had clients who have done animal studies already. And our guidance to them has been to use roughly about 5 grams per kilogram of each preconcentrate. And that's based off of the fact that the individual components have a high upper limit of 5 grams per kilogram. So it's possible that you could dose higher, but we don't have the data to support higher than 5 grams per kilogram. We're just going off of the individual components themselves.
Great, thank you. How would ABISOL work as a solubilizer for a clear gel formulation?
I think Chhanda should handle maybe that one.
Yes. So I think a part of it was also explained during the course of my presentation, that on diluting with water, the clarity is retained, the globule size is retained. And in this form, I mean, we can use it in a gel. And since water is present, you can use all the regular gelling agents which are used for thickening or topical gel. So it is very, very possible to use ABISOL in a topical gel formulation without compromising on the clarity of the end topical gel. So it can be very safely used and evaluated.
That's great. And can it be used to replace propylene glycol? You know, when we think about any costs for the formulation development.
Yeah, definitely. Yes, that's a very nice question. I mean, cost, yes, but then one has to see about the risk of the toxic impurities involved in solvents like propylene glycol and compare with that. So comparing that, definitely ABISOL works as a value added excipient, you know, that should be evaluated. So taking the risk of toxicity involved these days and the recalling of formulations which is happening across the globe, it would be worthwhile to evaluate ABISOL in place of propylene glycol. Yeah, from that perspective.
Thank you. And I think there was a question here about limits of ABISOL. I'm looking at question number 8 specifically, Dr. Kapadia and Dr. Bowers. Do you know the limits of ABISOL? Any limitations?
So I think that's referring to the inactive ingredients database. And the ABISOL kit, we do. So again, we haven't tested these in animals by themselves per se, but the individual ingredients are all GRAS. They're all used in products already out in the market. And we know the individual IID limits for these various excipients. So we could potentially put together a chart that would provide guidance based off of the concentrations of those IID values of those individual excipients. But to say that we have IID values specifically for the preconcentrates by themselves, we wouldn't, obviously, because these ABISOL kits have not been used in a commercial product yet.
Thank you.
Yes, I do agree with Heath here. And as Heath rightly pointed out, each of these ingredients in ABISOL are listed in the IID, and the safety profiles for each of these ingredients have already been established. So it's really safe to evaluate ABISOL.
Great, thank you. Are surfactants needed when using ABISOL?
No, the ABISOL kit contains two surfactants and an emulsifier, so you do not need to add additional surfactant unless you find that it improves the solubility of your API. However, I would caution to make sure that your emulsion stays stable when you're adding more surfactant. It's possible you may get multiple particle sizes and multiple particle populations as a result of increasing surfactant.
Thank you. Now, I know we talked about the inactive ingredients, and I see a question for IIG here. And I thought this was also a good lead in to ask if there's any other limitations that our audience should know about as we're getting close to having to wrap up the program today when using ABISOL.
Well, the key, and I'll, this is true for all lipid formulations, and Chhanda can also chime in here. You know, especially with SED systems, one of the biggest limitations with the SED system is the amount of drug that you can actually load into the SED systems. Now, I've seen in the literature, you know, upper, around 20% weight loading being kind of the upper limit. However, I have spoken with clients who've gotten 50% loading into their SED systems. So really the limits that you have with SED systems and how much you can actually dose into an animal is based off of how much solubility you can obtain in your SED system, as well as the ABISOL kits. Chhanda, maybe you have some additional input?
Yeah. So one thing which always comes to my mind is that whenever you are evaluating these surfactant systems, like for example with ABISOL also, we are trying to find out a correlation, you know, between the physicochemical properties of the API and the different ABISOLs, maybe in terms of the log P value, in terms of the melting point, in terms of the molecular weight. But at the end of the day, what finally works and finally what a formulator needs to do is the actual solubility studies, because nothing is going to replace these solubility studies, and these solubility studies will help us understand what is the solubility of the API in the ABISOL and actually how much can you load into a formulation. So that always becomes very important. Nothing can replace the solubility studies. So that needs to be done.
Thank you. And can ABISOL be used as an emulsifier to help with lowering HLBs in instances such as oils?
ABISOL itself is a preconcentrate that contains emulsifiers in them. If you had an oil, say your API was an oil, let's call it like fish oil, or something along those lines. You could potentially use an ABISOL kit to emulsify that oil. I can't imagine. I can't imagine a situation where you would use ABISOL to emulsify a solid API. It's really going to that would be solubilizing the API, not really emulsifying it. So I don't know, Chhanda, do you have?
Yeah.
input?
So I when I'm looking at the question, if the person who's put in the question, the idea is to evaluate it as a primary emulsifier for a topical formulation, like a topical cream and all, then really no, not in that category, but definitely as a solubilizer to solubilize an API in a topical formulation and also enhance the stability of a topical formulation. But as a primary emulsifier in a topical formulation, it may not function because that's not the role for which it has been, we have made ABISOL. So it may not work as a primary emulsifier, but as a very good solubilizer if one talks about a topical formulation.
Thank you. Again, we are getting close to nearing our hour, so we will have to begin to wrap up with I have a really great closing question, actually, that I'm excited to jump to in a second. But again, to our audience, don't be discouraged. The doctors will be reviewing your questions and responding to them offline. So I love thinking about future treatment. It's one of my favorite topics. And this final question that we're going to jump to today, can ABISOL help formulate some of these newer novel modalities such as PROTACs, cyclic peptides, RNA, DNA fragments?
Yeah, that's a great question. And the short answer is yes. We actually have customers doing this already with the ABISOL kit. I know at least two companies that are using ABISOL for PROTACs. I have in conversations with another customer who's looking at peptides. RNA DNA fragments is something new. I've not seen that yet with the ABISOL, with the folks that are actually doing that work yet. However, I wouldn't say no to that. I think there is definitely a way to look at RNA and DNA. It might take some tweaking a little bit, but I think there's definitely a way to incorporate RNA DNA into a SED system.
Great. Definitely look forward to learning more about that and see how that evolves moving forward. So we are wrapping up and we're at the top of our hour. Doctors, thank you so much for your time today, especially as we're broadcasting this live around the globe. It's really greatly appreciated. I also want to thank our audience for hopping on and listening to the doctor's presentation and for being so interactive today. A lot of great questions and still quite a few questions to keep the doctors busy once this broadcast concludes today. Most importantly, I'd like to thank both ABITEC and IMCD Pharmaceuticals for making this broadcast possible today. I will remind our audience that this will be available for replay. As this presentation concludes, we'd just like to ask our audience if they would just kindly participate in a very brief survey that'll auto-populate on your screen just after today's presentation concludes. We'd really greatly appreciate your insight toward our future programs. And then you'll also receive an email alerting you when this webcast is available for replay, and we kindly ask that you pass that on to any colleagues who may find today's presentation insightful. So again, thank you to everyone for joining, and we'll see you next time. Bye-bye.